But what is a cell? Let me ask you a question.
Yeah.
Darwin wrote The Origin of Species in 1859, published it in 1859. He had an idea of the cell as being quite simple, correct?
Yeah, everybody did.
Okay. If he thought of the cell as being a Buick, what is the cell now in terms of its complexity by comparison?
A galaxy.
If Darwin thought a cell was, say, a mud hut, what do we now know that a cell is?
More complicated than a Saturn V.
So what is in a cell as far as we know now?
A world that Darwin never could have imagined.
I needed someone who could give me a glimpse into this world. So we went to molecular biologist Doug Axe.
Think of a cell as being a nanofactory, a factory where, on a very small scale, digital instructions are being used to make the components of the factory. Here we have the famous DNA double helix. You can see the two helical strands that are intertwined and wind around each other on the outside of the molecule. This is the material that stores all of our genetic information. In higher life forms, this will be the equivalent of something like a gigabyte of information stored in the molecules that form the individual chromosomes, all packed within the nucleus, which is a tiny fraction of the entire cell size. So what does DNA do? Well, the information in DNA ends up providing the information for sequencing the amino acids to make protein. We have information in a one-dimensional form that provides the information for a three-dimensional form.
New York City is a pretty busy place. The traffic, the people, the messaging, the systems of transportation. But when comparing a living cell to this great city, New York pales in comparison. A cell is so complex that scientists have only scratched the surface of what really goes on in there. However, even with our limited understanding, what we do know is that the inner workings of a simple cell are simply extraordinary.
It's almost like there's a miniature city in every cell, and everybody has their own little job. If all the traffic lights decided to all just go crazy, you have issues that not only do we fix the light, but also we have traffic issues, we have crime issues, we have problems that snowball from one part breaking down.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a two-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperonin. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperonin achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
So, Paul.
Oh, right. Definition.
And anything else you would like to add, about what you're thinking about and how it relates to this session?
Okay. Well, first, to get this out the way, I think I like to think of life not in terms of the stuff of which it's made, which is what a lot of people working on prebiotic synthesis do. How do you do the chemistry? I've nothing against chemists, but I think that they've got a long way to go. I see the real secret of life is the way it processes information. If you talk to a biologist, they use the language of signals and codes and transcription and translation. It's all about information storage and management and processing. I guess it was Jim who was saying that, maybe it was Joshua, maybe you both said about the analogy between living systems and electronic circuits with logic components and wiring diagrams, and in many cases you can treat these things as a Boolean network. And so if you take the gene regulatory network, for example, for the cell cycle of yeast, is something we've been doing. Treat it as a Boolean network. You can look at information patterns, you look at things like transfer entropy, and you can work all that out. You can do these computations, and there are distinctive motifs. So life is managing information in a distinctive way. There's a very visionary paper. You'll have to stop me. I'm going to get into full flood. By Paul Nurse, the former president of the Royal Society, called Life, Logic and Information, published in Nature about 6 years ago, that foresees the future of biology as really a bit like electronics or computer engineering. And the interesting thing about that way of looking at it is a wonderful essay that some of you may know called Why a Biologist Can't Fix a Transistor Radio. Because the way biologists go about investigating their subject isn't really to say, here we have a component, what does it do, what is its function, what does it connect to? If you think of it like that, just like a radio engineer can fix a defective radio without knowing anything about how a transistor works. If, for example, you have a defective cell, might be a cancer cell, if you understand the topology of the gene regulatory network and the information flow, you might be able to fix it by rewiring it.
Well, in 2010, the noted microbiologist Craig Venter and his team accomplished an incredible feat, surpassing their previous accomplishment of determining the complete sequence of the human genome. They created the first computer-designed, synthetically produced genome, which is the set of application programs for an organism. This artificial DNA had over 1 million letters of genetic code that were then read, processed, and executed by the computer systems in the target cell's nucleus. Thanks to Venter, these biological computers are no longer theoretical, they are experimentally verified. In an interview, Venter stated, "Life is basically the result of an information process, a software process. Our genetic code is our software, and our cells are dynamically, constantly reading that genetic code." When cells were discovered over 300 years ago, it was thought that the structure of a cell was very simple. With the inventions of the electron and proton microscopes, we now have a much clearer understanding of the intricacies of life. The cell is an extremely efficient and well-networked computer system with thousands or even millions of processors and billions of bits of information. Cells are the structural and functional units of all living organisms. Some organisms, such as bacteria, are unicellular, consisting of a single cell. Other organisms, like humans, are multicellular, having many cells. In fact, it's estimated that an adult human has over 100 trillion cells, and each cell is an amazing world of its own. It can take in nutrients, convert the nutrients into energy, carry out specific functions, and reproduce itself as necessary. Even more amazing is that each cell stores its own set of instructions for carrying out each of these activities. Let's take a closer look at how a cell works.
The first thing we see is the cell membrane. Much like the firewall software on your computer, the membrane contains protein gatekeepers, allowing only those components into the cell that belong and rejects all other components. Once we pass through this membrane, we see the organelles, or the organs of the cell. Ribosomes are the computer-controlled protein manufacturers of the cell. By fastening to an mRNA and using it as a template, the ribosome arranges amino acids in the correct sequence to form a particular protein. This process is known as translation. Golgi body. Next, we find a Golgi apparatus, also known as a Golgi body. Found in both plant and animal cells, Golgi bodies package and store proteins. Much like a post office, the Golgi packages and labels items, which it then sends to different parts of the cell. It's composed of membrane-bound stacks known as cisternae. Each cisterna is made up of a flattened membrane disc, and its primary job is to modify proteins, but it's also involved in the transport of lipids around the cell and the creation of lysosomes. Lysosomes. Lysosomes are spherical organelles which dispose of all the waste in the cell. These tiny organelles contain acid hydrolase enzymes that break up waste materials and cellular debris. The membrane around the lysosome allows the digestive enzymes to work at the 4.5 pH they require to break down the debris and dispose of it. Without the lysosomes, the cell would become overwhelmed with debris and ultimately self-destruct. Mitochondria. Mitochondria are the power plants of the cell, generating most of the cellular energy known as adenosine triphosphate, or ATP. In addition to supplying ATP, mitochondria are involved in a range of other processes, such as signaling, cellular differentiation, cell death, the control of the cell cycle, and cell growth. Nucleus. The nucleus is the control center of a cell. This is where we find most of the DNA which contains the instructions needed to create other elements of the cells. Like the motherboard of a computer controlling its components, the nucleus directs the growth, metabolism, and reproduction of the cell, among other things.
Life requires fully functional DNA, RNA, ATP, enzymes, and other proteins. If any of those components were missing or not working properly, life would fail to exist. This continues to generate several unanswered questions. How could non-living material develop the hardware and software known to be required by all living organisms? How did nature develop the arbitrary protocols for communication and coordination among the thousands of computers in each cell? These kinds of questions lead to interesting discussions among scientists. Those who adhere to a purely naturalistic explanation for the existence of life are left struggling to find mechanisms to create such a sophisticated information source.
The cell really is like nothing we've ever seen in the physical world. That's got to be firmly grasped. That's not something we can just say, oh, well, it's just a little bit more of the same old, same old. It's not the same old, same old.
Now, I think one good place to start is with a quote from Ernst Haeckel. Ernst Haeckel arguably was one of the most famous evolutionists of Darwin's day, a cell biologist, an embryologist in his own right. Ernst Haeckel actually named the bacterial kingdom. He named the bacterial kingdom the kingdom Monera. It's since been split into two new kingdoms, Archaebacteria and Eubacteria, but he named the bacterial kingdom. And listen, I want you to see how Ernst Haeckel described the cell. Now, this is a quote from 1883. Darwin published The Origin of Species in 1859. So this is close to 25 years after Darwin published The Origin of Species. 25 years after that book was published, this is how they viewed the cell. And when this is your view of the cell, it's easy to imagine how a cell could form all on its own through purely natural process. Ernst Haeckel. The cell is not, he's talking about a bacterial cell here. The cell is not composed of any organs at all, but consists entirely of shapeless, simple, homogeneous matter. It's nothing more than a shapeless, mobile little lump of mucus or slime, consisting of albuminous combinations of carbon. It's nothing more than a shapeless little lump of mucus or slime. That's how he described the cell. Because they didn't know what the cell was back in Darwin's day. They didn't know what we know today. And so, it's easy to envision how a cell, if it's nothing but a shapeless little lump of mucus or slime, how that could form in a puddle of mucus or slime. if that's all it is. It's easy to see how something like that could form through purely natural processes. Now, he's talking about the bacterial cell here, but even the bacteria does have organs. They call them organelles. It has an important organelle called the ribosome and performs most of the other processes. It just doesn't have them sequestered in these little structures that other cells have that we call organelles. Cells do have organs. They have what we call organelles. And the bacteria is far from simplest little lump of mucus or slime. The bacteria, in fact, has one of the most complex molecular machines known to biology called the flagellum. This is an animation of the bacterial flagellum, an extremely complex rotary motor that's used by bacteria to swim through water. Extremely complex motor possessed by the bacteria. A shapeless little lump of mucus or slime, bacteria are not. That's what we have found in the bacteria. And Ernst Haeckel was actually a proponent of spontaneous generation. Spontaneous generation. The view that life could spontaneously generate in, if you had some peanut butter or some broth, chicken broth or something sitting out, we know if you leave chicken broth sitting out, eventually you're going to get some bacteria and fungus and all kinds of stuff growing in that. Back in, Ernst Haeckel's day, they believed that life just could spontaneously generate in these kinds of solutions. And Ernst Haeckel was a proponent of spontaneous generation. This quote from 1876, again, and advocating for spontaneous generation, but look at his justification. Listen to this quote. If we do not accept the hypothesis of spontaneous generation, then at this one point in history of evolution, we must have recourse to the miracle of supernatural creation. He believed in spontaneous generation because what's the alternative? Creation. And he can't accept that. This quote, 1876, in 1859, Louis Pasteur performed his famous experiment where he completely disproved spontaneous generation. He cooked up some beef broth and put it in a flask that had a bent neck called a goose neck, and proved that beef broth would stay free of bacteria and fungus forever. That those things were in the air, and if left exposed to the air, those bacteria and fungus would get in there. But if you have a beef broth positioned in such a way where bacteria couldn't fall into it, it would stay sterile forever. He disproved spontaneous generation in 1859. And here it is, 1876, Ernst Haeckel still arguing for the acceptance of spontaneous generation, because what's the alternative? Creation. Cell is very complex, and this is what evolutionists have to explain today, the tremendous complexity of the cell. In comparison to our marvels of modern technology, the cell has orders of magnitudes more moving parts. And I want you to listen to how an evolutionist of our day describes the cell. This is Michael Denton, evolutionist, who has turned into an advocate for intelligent design. In his book, Evolution, a Theory in Crisis, Michael Denton, evolutionist, former evolutionist, described the cell this way. He said, to grasp the reality of life, as it has been revealed by molecular biology, we must magnify a cell 1,000 million times until it is 20 kilometers in diameter and resembles a giant airship large enough to cover a great city like London or New York. What we would then see would be an object of unparalleled complexity and adaptive design. That's how we view the cell today, an object of unparalleled complexity. Back in Darwin's day, simple little lump of mucus or slime. Fast forward 150 years, an object of unparalleled complexity, meaning nothing we can point to parallels or equals the complexity that we see within the cell. Now, Michael Denton described it as like a giant airship. I guess if you're talking about a big sci-fi, spaceship or something, that might work, but something like an airplane just doesn't do it justice. What we're talking about is a city of complexity.
Well, then how did it get created?
Well, by a very slow process.
Well, how did it start?
Nobody knows how it started. We know the kind of event that it must have been. We know the sort of event that must have happened for the origin of life.
And what was that?
It was the origin of the first self-replicating molecule.
Right, how did that happen?
I told you we don't know.
So you have no idea how it started?
No, nor has anybody.
Nor has anyone else.
We need to discover what those informational motifs are, by looking at simple living things or components of living things, and then try to come up with a theory of the complexification of information management to see if there are principles of thresholds at which these things jump.
Living beings are further examples of open systems which can produce order if energy is supplied. When the sun is shining, green leaves convert carbon dioxide and water molecules into oxygen and complex carbohydrates. Disordered small molecules, representing high entropy, are transformed into ordered large molecules, a state of low entropy. Again, this entropy decrease would never take place even if the entire energy of the sun were available, unless a complicated photosynthesis machine pre-exists in the chloroplasts of the leaf. It must already contain the program and the information for the order to be produced when the energy is switched on. Nothing new emerges. Another category of entropy reduction by energy exchange is the formation of crystal structures when heat is exported out of an open system. Water molecules, for example, will slow down their disordered random movements when they are cooled. Due to the directed interatomic forces between them, they will at one temperature stick together and form a perfect hexagonal crystal lattice. It is energetically favorable for them to arrange in this way. The accumulation of many such hexagons results in the formation of a crystal with a six-fold symmetry, as we can observe it in the snowflakes. We must now ask, is it enough to export energy if we want to obtain such a complex order out of disorder? The answer is no. The symmetry of the H2O molecules and their directed interatomic forces must already exist before. Otherwise, the most extreme cooling process would never deliver an ordered structure. Therefore, the order information is already present before the process starts. The visible crystal symmetry is pre-programmed in the molecules, which simply move into their state of lowest energy, a state which is defined by the H2O geometry. No new order or information is produced by crystallization. The final category of order formation by energy exchange are the so-called dissipative structures. The photograph shows what happens in a pan of oil when it is heated above a certain temperature. Characteristic ordered cells emerge out of the disordered liquid, which are called Bénard cells. Further such examples are the vortex in a tornado, a standing wave in a flute, or oscillating chemical reactions. In all cases, energy is dissipated in a way that stable ordered structures appear. If we regard the cross-section image of the Bénard cell, we can explain the phenomenon. The heat energy from below is transported to the colder surface above via stable convection cycles in the oil. They are stable because an increase of their velocity would result in a larger energy transport. The upper surface would therefore get hotter. This, in turn, would mean a reduced temperature difference between the two surfaces. Since this difference is the driving force for the convection, it would slow down again. Then, however, the temperature difference would increase again and the convection would accelerate, and so on. With other words, any deviation from the stable cycle results in a force that drives the system back into the stable state. This is simply a resonance in a feedback loop. We finally arrive at the same conclusion as with the other examples. It is not enough to import energy. As a prerequisite, a feedback mechanism must exist before, a mechanical or physical arrangement that already contains the information for the order that will become visible at a certain point. In contrast to the categories of complexity discussed so far, biological order may be described as specifically complex order. That means such a complex order, like the DNA or a wing, which represents a specific functional or aesthetic organization. The important difference is that biological organs represent new information. This one is not pre-programmed in an ordering machine. It is also not pre-programmed in the geometry of the constituent molecules. That means it is not energetically favorable for molecules to arrange in the form of a living creature. Finally, it is not pre-programmed in a feedback loop either. Therefore, we are led to the following conclusions. The law of thermodynamics demands that material order always turns into disorder, and never vice versa. In open systems, the exchange of energy allows for certain categories of exceptional limited order formation, which is sometimes called self-organization. However, it does not organize itself. It emerges only if the structural information has been organized before the process starts. Such a machine, a molecular structure, or a feedback loop in which pre-existing information for specifically complex structures of biospecies is organized, does not exist. The hypothetic process of evolution claims that disordered molecules can eventually turn into specifically complex order. Therefore, this process does not belong to the open system exceptions. It is thermodynamically excluded. The result of this examination is that the evolutionary theory is in contradiction to the second law of thermodynamics, which is the basis of nature. Therefore, it does not belong to reality.
How does Darwin or Darwinism say life began?
Well, he didn't know, and in fact, nobody knows. So, Darwinism, strictly defined, starts after the origin of life and deals only with living things.
Well, how can there be a theory about life without a theory about how life began?
Well, a grand overarching evolutionary story, of course, does include the origin of life, but Darwin's theory doesn't begin until you have the first cell.
Well, does someone have a theory about how life began?
This is the story of a small planet in space called Earth.
For a typical Darwinian explanation of how life originated, Dr. Wells directed me toward this documentary.
The chemical elements essential for life, hydrogen, oxygen, carbon, and nitrogen, were now in place. What was needed was a way of combining them. Perhaps the energy came from lightning. Whatever it was.
Excuse me?
Whatever it was, energy managed to arrange these chemical ingredients in just the right way.
Whatever it was? I was hoping for something a little more scientific. The most popular idea has been that life emerged spontaneously from primordial soup. In 1953, Stanley Miller mixed water, methane, ammonia, and hydrogen to simulate the early Earth's atmosphere. Then he ran electricity through it in an attempt to jumpstart life. It didn't work. While the initial results seemed promising, 50 years later, most serious scientists have abandoned this approach in favor of alternate theories. Prominent Darwinist Michael Roos attempted to explain one of them to me. He wasn't kidding. How did we get from an inorganic world to the world of the cell?
Well, one popular theory is that it might have started off on the backs of crystals.
My crystal ball.
Molecules piggybacked on the back of crystals forming, and that this led to more and more complex. But of course, the nice thing about crystals is every now and then you get mistakes, mutations, and that this opens the way for natural selection.
But at one point there was not a living thing,
yeah.
And then there was a living thing. How did that happen?
Well, that's just I've just told you. And I don't see any reason why you shouldn't go from very simple to more and more complex to more and more complex.
I don't either, but I don't know how you get from mud to a living cell. That's my question.
Yes, well, I've told you.
I think it was on the back
try
of
one more
crystals.
time.
On the backs of crystals.
On the backs of crystals is at least one hypothesis, yes.
So that's your theory, and you think that is more likely and less far-fetched than intelligent design?
I think it is.
I wouldn't put Ben Stein's money on Dr. Roos's joyriding crystals, but it did make me wonder, what were the chances of life arising on its own?
It's been speculated that probably there would have to be a minimum of about 250 proteins to provide a minimal life function. If that's really true, then I think it's almost inconceivable that life could have happened in some simple step-by-step way.
Okay, so the simplest form of life requires at least 250 proteins to function. What's so difficult about that?
Welcome to the Casino of Life. Who wants to spin for a chance to win?
Oh, sure. I'll give it a shot. What do I win?
Take a look at this.
Huh? How about the world's first single-cell organism? This perfectly aligned string of proteins could be yours.
Now, take a spin.
I won!
Tina, tell him how many times he needs to do that to win the prize.
250.
That's right, folks, and all in the correct order.
But that's impossible.
We've heard that before, haven't we, Richard?
Come on, Mother Nature, do your thing. You stupid machine! I hate you!
We're talking about something that's staggeringly improbable, roughly 1 in a trillion, 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 trillion.
Let all of life choose a million, a trillion, trillion. The number is essentially zero. Something has to skew nature to choose the ones that work.
So, in the game of life, it looks as if the house always wins.
Karl Popper also makes the argument. Now, Karl Popper is best known for his contribution to the scientific method, what's called Popperian falsification. Remember, I described the scientific method. You start with the hypothesis, and then a theory, and then you try to falsify your theory. This is a part of the scientific method that's attributed to Karl Popper. Listen to Karl Popper. He says, what makes the origin of life and the genetic code a disturbing riddle is this. The genetic code is without any biological function unless it is translated. That is, unless it leads to the synthesis of proteins whose structure is laid down by the code. But the machinery by which the cell translates the code consists of at least 50 macromolecular components which are themselves coded in the DNA. Thus, the code cannot be translated except by certain products of its translation. He continues, this constitutes a baffling circle, a really vicious circle, it seems, for any attempt to form a model or theory of the genesis of the genetic code. Thus, according to Karl Popper, we may be faced with the possibility that the origin of life, like the origin of physics, become some impenetrable barrier to science and a residue to all attempts to reduce biology to chemistry and physics. That's powerful. That is a powerful admission from someone like Karl Popper, because that's where we are today. They just have nothing that they can really put on the table to explain how a cell formed all on its own through purely natural processes.
And what's fascinating to me, not only do proteins create proteins, but we have proteins that are actually creating the DNA molecule. That's our blueprint. And yet on the DNA molecule, there are sequences, a part of the DNA that are coded for those proteins. So the manufacturing plant needs the blueprint, and the blueprint, the DNA, needs the manufacturing plant in order to actually create the DNA. So what came first, the proteins that made the DNA or the DNA that made the proteins?
Darwin was trying to explain how the branches on the tree of life originated. He was trying to show how natural selection could have modified existing organisms to produce the great diversity of plant and animal life that fills the earth today. But when it came to the base of the tree, which represented the origin of the first life, the first living cell, Darwin had very little to say. In fact, in The Origin of Species, he didn't even address the question of how life might have originated from non-living matter.
The only glimpses we have of Darwin's opinions on the subject appear in a letter he wrote to a colleague named Joseph Hooker.
Regarding the first production of a living organism, if, and oh what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat and electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes, at the present such matter would be instantly devoured. But this may not have been the case before living creatures were formed.
During the final years of his life, Darwin did little to develop his idea that a primitive cell might have emerged from simple chemicals in the primordial waters of the early Earth. But later in the 1920s and 30s, a Russian scientist named Alexander Oparin formulated a detailed theory about how this could have happened. It was called chemical evolution.
Oparin thought that he could explain the origin of the first life using Darwinian principles. He envisioned simple chemicals combining and recombining to form larger molecules, and then these larger molecules organizing themselves with the help of chance variations and natural selection into the first primitive living cell.
Over the next 3 decades, many scientists worked to develop and refine these ideas as they pondered the questions both Oparin and Darwin had raised. How could life have evolved from simple chemicals? One man thought he knew.
The problem of biological origins has, for a very long time, I would say, has been a real deep interest to me just because of the scale of the problem, the importance of it. Where did we come from? Why are we here? All that kind of question. Probed from the point of view of natural science.
During the late 1960s and throughout the 70s and early 80s, Dean Kenyon was one of the leading chemical evolutionary theorists in the world. And like others in his field, he was trying to explain how life on Earth began through a purely natural process.
In 1969, Kenyon co-authored an important book on the origin of life.
Gary Steinman and myself thought that if we were to pull together all of the lines of empirical evidence that had accumulated by the mid to late 60s in one continuous argument, we were very enthusiastic about the possibilities for explaining the origin of the main life-building elements.
Despite his optimism, Kenyon faced a significant problem. To explain how life began, he first had to account for the origin of the essential building blocks of every cell that has existed on Earth. Large, complex molecules called proteins.
Proteins have a wide range of function in the cell, everything from structural requirements in terms of scaffolding of the cell, the cytoskeleton, to enzymes, where they're actually processing
molecules to harvest energy or to build components of the cell.
Proteins do pretty much all of the jobs inside of the cell, except for storing genetic information. That's left to the DNA, the RNA. But all the day-to-day jobs, cleaning up the cell, making energy, it's all proteins.
Kenyon knew that proteins would have been as important to the first life as they are to living cells today. He also recognized the complexity of their construction.
By the 1960s, scientists had determined that even simple cells are made of thousands of different types of proteins. And the function of these molecules derives from their highly complex three-dimensional shapes. The irregular shapes of some proteins allow them to catalyze or trigger chemical reactions because of the hand-and-glove fit that they have with other molecules in the cell, while other protein molecules form interlocking structural components.
The individual parts of a bacterial motor, like this ring structure, are each made of either a single protein molecule or an assembly of proteins fitted together into a specific shape. These proteins are, in turn, made of smaller chemical units called amino acids that are linked together in long chains.
There is a very great degree of intricacy of architecture down in the cell units in these protein-forming amino acids.
In nature, 20 different types of amino acids are used to construct protein chains. Biologists have compared them to the 26 letters of the English alphabet.
Alphabetic letters can be arranged in a huge number of possible combinations, and it's the sequential arrangement of the letters that determines whether you have meaningful words and sentences. If the letters are arranged correctly, you'll get meaningful text. But if they're not arranged correctly, you'll get gibberish. And the same principle applies for amino acids and proteins.
There are at least 30,000 distinct types of proteins, each made of a different combination of the same 20 amino acids. They are arranged, like letters, to form chains, often hundreds of units long. If the amino acids are sequenced correctly, then the chain will fold into a functioning protein.
Proteins are arranged with their amino acids in such a way that the amino acids collapse on each other into an architecture that is pre-programmed by the order of the amino acids. It folds into a certain structure, and that structure can do a certain function. So all proteins in the cell have a certain three-dimensional pattern that's based on the arrangement of amino acids in the chain.
This arrangement is critical, for if the amino acids are incorrectly sequenced, a useless chain forms, and instead of folding into a protein, it will be destroyed in the cell.
Proteins, like written languages or computer codes, have a high degree of specificity. The function of the whole depends upon the precise arrangement of the individual parts.
But what produces the precise sequencing of amino acids that gives rise to the specific shapes and functions of proteins? During the 1950s and 60s, discoveries about protein structure forced biologists to confront this mystery. Dean Kenyon believed he could solve it. In his book, Biochemical Predestination, Kenyon and his co-author, Gary Steinman, proposed an intriguing theory. Kenyon wrote, "Life might have been biochemically predestined by the properties of attraction that exist between its chemical parts, particularly between amino acids in proteins."
At the time that biochemical predestination came out, I and my co-author were totally convinced that we had the scientific explanation for origins.
Kenyon proposed that the chemical properties of the amino acids caused them to be attracted to each other, forming the long chains that became the first proteins, the most important components in the living cell. And this meant that life was effectively inevitable, predestined by nothing more than chemistry.
Many scientists embraced Kenyon's ideas, and over the next 20 years, Biochemical Predestination became a best-selling text on the theory of chemical evolution. Yet 5 years after the book's publication, Kenyon quietly began to doubt the plausibility of his own theory.
It was during that whole period of time that my doubts about certain aspects of the evolutionary account became apparent. When coming into contact with a powerful counter-argument given to me by one of my students, and I could not refute that counter-argument.
Kenyon was challenged to explain how the first proteins could have been assembled without the help of genetic instructions. In living cells today, chains of amino acids are not formed directly by forces of attraction between their parts, the scenario Kenyon envisioned on the early Earth. Instead, another large molecule within the cell stores instructions for sequencing the amino acids in proteins. It is called DNA.
Initially, Kenyon believed that proteins could have formed directly from amino acids without any DNA assembly instructions. And that's why so many scientists were excited about his theory. But the more he and others learned about the properties of amino acids and proteins, the more he began to doubt that proteins could self-assemble without DNA.
In DNA, Kenyon encountered a molecule with a property he could not explain through natural processes. For locked securely within its double helix structure is a wealth of information in the form of precisely sequenced chemicals that scientists represent with the letters A, C, T, and G. In a written language, information is communicated by a precise arrangement of letters. In the same way, the instructions necessary to assemble amino acids into proteins are conveyed by the sequences of chemicals arranged along the spine of the DNA. This chemical code has been called the language of life, and it is the most densely packed and elaborately detailed assembly of information in the known universe.
Like other scientists working on the origin of life, Kenyon realized he had two choices. Either he had to explain where these genetic assembly instructions came from, or he had to explain how proteins could have arisen directly from amino acids without DNA in the primordial oceans. And in the end, he realized he could do neither.
It's an enormous problem how you could get together in one tiny submicroscopic volume of the primitive ocean all of the hundreds of different molecular components you would need in order for a self-replicating cycle to be established. And so my doubts about whether amino acids could order themselves into meaningful biological sequences on their own without pre-existing genetic material being present just reached, I guess for me, the intellectual breaking point sometime near the end of the decade of the 70s.
As Kenyon reevaluated his theory, new biochemical discoveries further weakened his conviction that amino acids could have organized themselves into proteins.
The more I conducted my own studies, including a period of time at NASA Ames Research Center, the more it became apparent that there were multiple difficulties with the chemical evolution account. And further experimental work showed that amino acids do not have the ability to order themselves into any biologically meaningful sequences.
Faced with mounting difficulties in his own theory and a growing body of scientific data about the importance of DNA, Kenyon was forced to confront the absolute necessity of genetic information.
The more I thought about the alternative that was being presented in the criticism and the enormous problem that all of us who worked on this field had neglected to address, the problem of the origin of genetic information itself, then I really had to reassess my whole position regarding origins.
For Dean Kenyon, a new question became the focus of his search for life's origin. What was the source of the biological information in DNA?
If one could get at the origin of the messages, the encoded messages within the living machinery, then you would really be on to something far more intellectually satisfying than this chemical evolution theory.
Yet Kenyon realized that he faced a narrowing set of options. By the 1970s, most researchers had rejected the idea that the information necessary to build the first cell originated by chance alone. To understand why, consider the difficulty of generating just 2 lines of Shakespeare's play Hamlet by dropping Scrabble letters onto a tabletop. Then consider that the specific genetic instructions required to build the proteins in even the simplest one-celled organism would fill hundreds of pages of printed text.
Of course, serious origin of life biologists did not believe that life had arisen by chance alone. Instead, they envisioned natural selection acting on random variations among chemicals to produce the first life. But there was a problem with this proposal.
By definition, natural selection could not have functioned before the existence of the first living cell. For it can only act upon organisms capable of replicating themselves, cells equipped with DNA that pass on their genetic changes to future generations.
Without DNA, there is no self-replication. But without self-replication, there is no natural selection. So you can't use natural selection to explain the origin of DNA without assuming the existence of the very thing you're trying to explain.
Chance, natural selection, and his own theory of self-organization had all failed to explain the origin of genetic information. Now Kenyon saw only one alternative.
We have not the slightest chance of a chemical evolutionary origin for even the simplest of cells. So the concept of the intelligent design of life was immensely attractive to me and made a great deal of sense, as it very closely matched the multiple discoveries in molecular biology.
In the years since Kenyon's rejection of chemical evolution, science has revealed the details of an entire system of information processing that bears the hallmarks of intelligent design.
First, we start with our DNA double helix. Helicases are molecular motors that use the chemical energy of ATP to break hydrogen bonds between bases and unwind the DNA double helix into single strands. This process is used for both replication and repair of the strands. If unwound, a single DNA strand would be about 6 feet in length. And yet, it fits into a cell which can't even be seen with the human eye. Remarkably, these strands are kept from tangling as they are separated and duplicated. Next, these strands are copied by enzyme-based computers, creating an exact replica of each original strand. Over 30 different proteins are required for duplication and repackaging of cellular components during cell division.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.
A couple other quotes before I close out. Klaus Does is the director of the Institute of Biochemistry at Johannes Gutenberg University in West Germany. And Walter Bradley in The Case for Faith, I don't know if you read it, awesome book, I'd recommend it, refers to Klaus Does as the biochemist who's considered one of the foremost experts in this area. Klaus Does, more than 30 years of experimentation on the origin of life in the fields of chemical and molecular evolution have led to a better perception of the immensity of the problem of the origin of life on Earth rather than to its solutions. At present, all discussions on principal theories and experiments in the field either end in stalemate or in a confession of ignorance. Paul Davies is a professor at Arizona State University and as well is the director of Beyond and was previously the chair of the SETI program, the Search for Extraterrestrial Intelligence. Paul Davies, it's a shame It's a shame that there are precious few hard facts when it comes to the origin of life. The how part has everyone stumped. Nobody knows how a mixture of lifeless chemicals spontaneously organize themselves into the first cell. And he admits, said many investigators feel uneasy about stating in public that the origin of life is a mystery, even though behind closed doors they freely admit they are baffled. Why? They worry that a frank admission of ignorance will undermine funding.
Could you make a distinction between what you just said and, say, computer architecture? What happens artificially with man-made technology?
I think there is a lot. It's a very close analogy. Of course, we've designed computers precisely to fulfill these functions. Mm-hmm. If I'm giving a lecture on this stuff, I usually show a picture of a computer with a screenshot, and I say, to me, just like life looks like magic, Windows looks. Sorry about Mac users. Windows looks like magic. Give me an explanation, and then I show a picture of the innards of the computer, and arrows pointing to silicon and copper and iron and so on. And a computer engineer might well tell you, and might say, well, it's sort of complicated in there, but it's got something to do with the silicon, and we're hot on the trail, and we're trying to figure it out. We need to get down to a smaller scale and work out what all that little patterning is, and it's really complex. But give us enough money, and one day we'll work that out. And that that's not a full explanation. You've got to go and see a software engineer and be told it works because of this code and that code, and so that's the software story. So I think the two are actually really very close.
Yeah, more and more now we're seeing from scientists statements that design is obvious in living things. Right. For example, D.L. Abel, writing in the International Journal of Molecular Sciences, says to stem the growing swell of intelligent design intrusions, it is imperative that we provide standalone natural process evidence of non-trivial self-organization at the edge of chaos. We must demonstrate on sound scientific grounds the formal compatibilities and naturally occurring physiocodynamic complexity. Did I say that right? It's not a word you use every day. He continues, at the same time, we have spent much of the last century arguing to the lay community that we have proved the current biological paradigm. Unfortunately, he says, very few in the scientific community seem critical of this indiscretion. One would think that if all this evidence is so abundant, it would be quick and easy to falsify the null hypothesis put forward above.
That's right. And then he says science has an obligation to be honest about what the entire body of evidence clearly suggests. We can't just keep endlessly labeling abundant evidence of formal prescription in nature apparent. The fact of purposeful programming at multiple layers gets more apparent with each issue of virtually every molecular biological journal.
Since the late 19th century, since the time of Darwin, in fact, in part because of Darwin's writing in The Origin of Species, scientists came to accept a convention, a definition of science that excluded the possibility of design as a scientific explanation. And that convention has a name. It's called methodological naturalism. And it just means that if you're going to be scientific, you must limit yourself to explanations that invoke only natural causes. You can't invoke intelligence as a cause.
But I don't want a designer for life, you see. So I want Mother Nature to somehow contrive this without literally wiring the thing.
And yet, curiously, we make inferences to intelligence all the time. It's part of our ordinary reasoning to recognize the effects of intelligence.
Consider, for example, these hieroglyphic messages carved upon the ruins of Egyptian monuments. No one would attribute the shapes and arrangements of these symbols to natural causes like sandstorms or erosion. Instead, we recognize them as the work of ancient scribes, intelligent human agents. Similar reasoning leads us to conclude that the mysterious stone figures on the shores of Easter Island were not formed by the actions of wind and water over great periods of time. Nor do we presume that plants could grow into these familiar shapes without some manner of intelligent guidance.
Of course, we make these inferences all the time, and we know they're correct. But the question is, on what basis do we make these inferences? What are the features that enable us to recognize intelligence?
The ability to detect information in electromagnetic transmissions has made possible a unique search for intelligence. For more than 3 decades, astronomers involved in SETI, the search for extraterrestrial intelligence, have monitored radio signals from outer space in an attempt to find information-rich patterns. Typically, radio telescopes receive either random noise or simple repetitive signals produced naturally by stars, galaxies, and other celestial objects. But astronomers recognized that if they ever identified an information-bearing signal, it would confirm the existence of intelligent life beyond the Earth. Some have speculated that an extraterrestrial civilization might have attempted to communicate by transmitting messages in the universal language of mathematics, perhaps through a recognizable pattern like a series of prime numbers.
You're not going to get that by chance. So you need complexity or improbability, lots of prime numbers, and you also need a pattern. And it has to be the right sort of pattern. It's not a pattern that you're imposing. It's a pattern that's there objectively.
To date, SETI research has failed to detect any pattern or information that would indicate intelligence in a distant galaxy. But in another universe, much closer to home, scientists have discovered a wealth of information within the nucleus of the living cell.
Bill Gates has said that DNA is like a computer program, only much more complex than any we've been able to devise. And if you reflect on that even for a minute, it's a highly suggestive observation, because we know that Bill Gates does not employ wind and erosion or random number generators to generate his software. Instead, he employs intelligent engineers, software engineers. And so everything we know in our experience suggests that information-rich systems arise from intelligent design. But what do we make of the fact that there is information in life, in every living cell of every living organism? That's the fundamental mystery. Where does that information come from?
And as we look around us at the millions of plant and animal species, it becomes incredibly breathtaking to ponder how much information and organization must take place for all of this life to exist.